I'm really pleased to have Rick Bostock here today. He's a distinguished professor in the plant pathology department at UC Davis. I got to know Rick when I was a student there and be a student of his during that time. His research has focused on plant diseases caused by fungal and oomycete pathogens. And his host plants that he's worked with have largely included crops like tomato, pepper, orchard crops, and nursery ornamentals. His research interests have generally been mechanistic, with a strong emphasis on plant microbe interactions and plant immunity. He teaches Introduction to Plant Pathology, graduate level biochemistry and molecular biology of plant microbe interactions, and a well-attended undergraduate science and society class on feeding the world. Rick is also an excellent fingerstyle guitar player and close to retirement. Are you retiring soon, Rick?
Yeah, July 1.
Oh, yeah. Wow.
It'll be 40 years with UC Davis. So,
wow.
Yeah.
Great.
So, yeah.
You know, I really appreciate that you have taken the time to come and offer us a class, a presentation on plant defense mechanisms.
Yeah, plant defense part 1, induced resistance, disease resistant plant activators. What plant activators are compounds that both natural and synthetic that can induce this in plants. So, as many, I think that most of you probably are, plants do have ways of defending themselves against pathogens and insects. There are morphological and chemical barriers. Bark would be a morphological barrier. It's kind of tough to get through bark, and that's why wounds are often needed. Certainly, certain flower structures and other structures on plants can prevent insect attack, or retard insects at least. There are chemical barriers, like in the peel of onions, red onions. There's a compound called protocatachoic acid, which can help resist against things like onion smudge disease. And then the way I like to think about this, there are preformed and inducible, but what I mean by preformed, they're already there. And the example of protocatachoic acid would be one. Nicotine is another that can be preformed, or it can be induced as well. And but what I mean by induced is these are defenses that are induced after the plant is attacked. Okay, and so the advantage of that is that they're formed at the right place at the right time. Basically, is the way we like to say it. And these can actually be chemical barriers as well. But there are some structures too that can be more of a barrier that helps resist ingress of like lignin, induced lignin, which can be pretty tough for a pathogen to get through. And these can be localized, or said by localized, I mean these defenses are induced at the site of attack, or they can be the plant can be alerted systemically, such that these defenses can be manifested somewhere else in the plant other than at the leaf or the site of attack. And so we call these systemic responses. And that's what I'm going to focus on today with induced resistance. So these are inducible, and some of it's localized, but I want to focus on the systemic responses. And there's been tremendous interest in induced resistance. And as you can imagine, it's my analogy with the immune response that we have in vertebrates. The idea is that you're harnessing natural plant defenses, and in this way, getting some control over the diseases and pests that attack our plants. So the way I like to organize it, I like to talk about systemic induced resistance, and there are a couple of different types. And so I'm going to try to show this to you on this little graphic here. We can think of induced systemic resistance to insects. We call it ISR, and some pathogens. And what's going on here, so a plant leaf here the lower leaf is being, can be attacked by an insect. Wounding to some extent can do this. Certain pathogens can do this as well. Not all, but certain pathogens. And there are root colonizing bacteria that can also do this. And what happens is that there, and then I should also add that a pathogen or a lister on this leaf can induce this. And so what happens in these upper leaves is they're protected against these attackers. And this is a generalized resistance. In other words, it's say an insect a particular insect species is damaging a plant, it can protect against a different species of insect. The same with pathogens. If a certain species of bacterium or a fungus is causing damage on one part of the plant, it can protect other parts of the plant against attack by the same pathogen or a different one as well, a different species. So it's a generalized resistance that's triggering the plant. And so again, induced systemic resistance is one type. And then the other type is systemic acquired resistance. And the kinds of signaling that goes on. By signals, I mean the chemistry that's underlying that's orchestrating these responses is a little different between these two types of resistances. Okay, does that make sense? Okay. Okay, so this goes way back, as I said. There's been a tremendous amount of interest in induced resistance, well over 100 years. And but where it really took off is about 60 years ago. And I wasn't in graduate school then, but but about 60 years ago, Frank Ross at Cornell did some very interesting work with the tobacco mosaic virus in tobacco, using that as a model system. And what he was able to show is that if you inoculated a lower leaf, excuse me, if you treated a lower leaf with water, or you treated it with TMV, the virus, and then you waited 7 days, and then you came back and challenged the upper leaves with TMV in this case, you could see that in the control, there's no protection against the necrosis that's being induced by this in this particular variety of tobacco. But the prior inoculation, this protecting inoculation, protects the upper leaves against TMV. And so a tremendous amount of work was done on this, and then they could show that, again, this idea of different types of pathogens could do very similar things by protecting against subsequent challenge. The lab I worked in as a graduate student many years ago, more than 4 decades ago, this was a major thrust of study. And what colleagues of mine did in that lab, they could show in cucumber, which was a good, again, we scientists like to think of model systems in order to show things experimentally. But in cucumber, challenged by the anthracnose fungus, Colotatricum laginarium, you could show this same phenomenon. And you know, they could do this in the field, in greenhouse, you know, wherever. What they did here, a lower leaf, which you don't see in this picture, was treated with water. And then about 4 or 5 days later, it was challenged by with the fungus. And you can see all these little dots is where they applied some spores. And you can see that there's no protection of this leaf. But if you hit that lower leaf with Colotatricum laginarium, and then waited 4 or 5 days, and then came back and challenged this upper leaf, you had no disease. Okay, and you have to wait, you know, a few days to see the disease develop. But there was no disease developing in this upper leaf. Okay. So systemic induced resistance, and we'll just talk about it generically. I know I kind of split it up between ISR and SAR, but systemic induced is the general term I like to use for this phenomenon. It's something that doesn't depend on direct killing of the pathogen or the pest, because as I said, some insects can be resisted by these mechanisms as well. But it has to do with increasing plant defenses to resist the attacker. It's a generalized resistance responses, and it requires induction of new gene expression. What I mean by that is that these leaves, the plant is kind of going into an alert, basically, and it's being triggered to be on the lookout, essentially, for new attacks. And so there's a change in gene, there's a change in chemistry of the plant as well. And then this resistance, what has been found, in addition to being biologically induced, like I just showed you with pathogens and pests, but it can also be induced by certain chemicals, both natural, which is of interest to organic growers, as well as synthetic, synthetic chemistry, which, and we call these, well, some of these we used to call elicitors. Now we tend to call them plant activators, at least the commercially. And you're probably aware of some of these. And I'll talk about some of these later on in my presentation. The other thing I want to get across is between these two types, there are distinct signals that are generated by these two different types of induced resistance. And so there's this fine regulation that has to go on in the plant between these phytohormone stress pathways, and it depends on the type of attacker. And so let me show you what those are. So in induced systemic resistance, what happens, you get this attack at the lower leaf. There's local wound responses that are created. There's some signaling, and some of the signaling we really don't know that much about. We've tried, but it's still kind of obscure in this case. But we do know that elsewhere in the plant, you get the increase in this phytohormone called gismonic acid. And gismonic acid then is a phytohormone that affects a lot of different pathways in the plant, such that you trigger the expression of these defense responses. On the other side of the plant, using my model plant here, certain kinds of pathogens or elicitors on the lower leaf, that will lead to lesions and local responses. So this leaf is pretty much lost to the cause. It's sort of the sacrificial lamb. But from that leaf, though, are signals that are generated. We know a little bit more about this in the case of systemic acquired resistance, what some of those signals are. And I won't get into those. But what we do know is that this causes an increase throughout the plant of salicylic acid, which is another very important phytohormone. And the salicylic acid then orchestrates a whole series of defense responses in those leaves. Okay. Now, so here's the model, and you can see that so when a plant and a pathogen or an insect pest interact, there's all kinds of signals that are generated at the site of attack. And we call this signal response coupling, such that there's an activation of defense genes expression and a whole slew of biochemical reactions. Now, what some of these things are, cell walls of the plant, the plant cell wall can become stronger. And so in some places you may get deposition of new cellulose and callose, which is a glucose polymer, let's just put it that way. And lignin, which is a really, as I said, and these induced lignins are really tough. When a cell wall is lignified, it can't get through. Like the shot holes that you see on some diseases, those are zones of lignin around those shot holes. And so that's essentially a defense response, if you will, even though we don't like to see shot holes on our fruit or on our leaves. Basically what the plant is doing, it's preventing further spread of the pathogen by simply walling off that area, and those dead lesions then fall out to give you the shot hole appearance that you see in a number of different diseases, for example. And then there are these antimicrobial compounds in proteins. Some of these we call phytolexins, antiviral compounds. peroxidases, and other proteins and so on that help restrict a pathogen from moving through the plant. Now, something that's really important here about how this all works is that it's not necessarily that all these things are produced at once, because if that were the case, it probably would not be a good thing for the plant, because some of these antimicrobial compounds that are produced are actually bad for the plant. They'll kill plant cells as well. They're phytotoxic. So what I want to get across to you here is this concept of priming. What a lot of these plant activators, for example, in induced resistance, they prime the plant. Basically, they crank up the surveillance system in the plant, alert it such that when it is attacked, it very rapidly responds to the attacker at the site of attack. And this is from, again, that cucumber and anthracnose disease. And if you were to look at one of these sites where we tried to get the infection going in this plant that was protected, you might see something like this. And so, in fact, you would see something like this. This is actually from a cucumber plant from a colleague of mine who did this many years ago. And what you see is at where a fungal spore, a fungal infection structure is, you see this zone of lignification right around that site of attempted. And so you don't want the plant to lignify, because that would be bad. But what you do want is for the plant to respond just where it needs to be at the right time, at the right place, at the right time, and to do it, very effectively. And lignin, all these cells around here, in this area, have become lignified, and so the pathogen can't get through that. And so we see this commonly with some of these other defense responses that I alluded to in the previous slide. So priming is really important here. So I know you're not chemists and all that, but this is what the structures of salicylic, very simply, it's very similar to acetylsalicylic acid, aspirin. So we like to think of salicylic acid as sort of like plant aspirin. And then gismonic acid is derived from the common plant fatty acid, linolenic acid. Okay, it's just an oxidized, metabolized version of it. But both of these have very, very profound effects on the plant. They, as I said, they function essentially as plant hormones and mediate all that. There are some synthetic compounds, with really the granddaddy of them all was acebenzolol or Actigard. This was really one of the first plant activators. It's originally developed by Ciba Geigy, which is now Syngenta. So again, this is not something that could be used in organic production systems, but it is used in conventional and then some other compounds. And these are both SAR inducers. That is, they operate by engaging salicylic acid pathways. So when you hit a plant with this, this is getting into the weeds a little bit, no pun intended, but if you look at the time course of how this develops in the plant, so these are not fungicides, by the way. When we trigger, like with these plant activators, you got to give them time to work. And this is shown with biological induction, in fact, very similarly. So if TMV, the tobacco mosaic virus, on this third leaf here, and then if you follow what happens to salicylic acid during the development of the induced resistance and all these other leaves. And so these are the leaf numbers here. And so it takes 96 to 144 hours after this initial inoculation on leaf 3. You see a big boost in salicylic acid in that leaf, but you begin to see it build up in leaves, particularly above the inducing leaf here. And you also get some in the roots. So acid, accumulates or builds up in the plant really throughout the plant, but it's most pronounced in these really top leaves of the plant. And so those would be the leaves that would have the strongest protection. They would have the strongest. Now, I have to say a little bit about gismonic acid. I'm not going to say a lot because most of these plant activators that I'm going to tell you about in a minute here primarily target the salicylic acid pathway. But this shows something really interesting too, as well. So again, a gismonic acid, we don't really know what the systemic signal is, but we know that gismonic acid builds up following like an insect attack in these upper leaves. And some of that resistance is due to some interesting proteins that the insects that try to attack these leaves and chew on them, they can't digest the proteins very well in the leaf tissue and basically starve and don't do nearly as well. But what's cool about this stuff, well, cool from a scientist, because we can make mutants and do some crazy things in the lab. If you have a plant that can't make salicylic acid, for instance, so you block salicylic, those plants become super susceptible to pathogens. So that just kind of affirms that salicylic acid is pretty important in the defense. And the same with gismonic acid. You can make plants that can't respond to gismonic acid. And you can see, well, here's the normal plant, which has been challenged by the spider bite, and it looks pretty good. But if you have a plant that can't respond to its own JA, it's gismonic acid, they become super susceptible or super vulnerable to insect attack. So they're pretty, so SA and I'm sorry, I go back and forth between the jargon, but salicylic acid and gismonic acid are really important just as part of the plant's natural defense repertoire, if you will. All right, so let's talk about plant activators. And as I mentioned, Actigard is probably the granddaddy of it all, developed many years ago, actually, back in the late 70s. This operates through the salicylic acid pathway. It's registered in the US on various coal crops, leafy vegetables, tomatoes, and tobacco. This is something we did some years ago in my lab where it works reasonably well against bacterial speck in tomato, and I understand it is used in greenhouse tomatoes. Yeah, bacterial diseases, we often don't have that much in our arsenal to use. Again, this is conventional stuff, but you can see how it protects against bacterial speck disease there. There's plant activators have been used to a large extent in rice blast disease control. At least at one time, they were the dominant chemicals that were used in rice blast control. This is probenazole or rhizomate. I don't think it's been used here in North America much. This is primarily in Japan. The interesting thing about rhizomate is the active part of it breaks down to form saccharin, the artificial sweetener. You remember, like Fresca? Okay. And so go figure. So saccharin can trigger this response, the induced resistance in rice, which I have always found very interesting. But there are other products too out there. Thiodinyl is a more recently developed product that is used with something that we worked with a few years back and published a paper on. But so these compounds are used at big time. Again, this is conventional, and these are, a synthetic chemistry here that we're dealing with. But both of these operate through the salicylic acid pathway. We sometimes call them salicylic acid mimics. And so what happens, so when you spray a plant with these, again, to emphasize, is it basically short circuits things a little bit. So instead of trying to do it biologically with a pathogen that causes a lesion on a leaf, you actually immediately get the chemistry going and turning on the gene expression that you need in order to produce these proteins that can make the various defense responses. Okay. Okay, so this gets us, and there's some other interesting products that have been developed. Regalia used to be called Milsana. When Marone Bioinnovations picked it up. They renamed it to Regalia. This targets various fungal bacterial diseases. It seems to have some efficacy against powdery mildew and gray mold. I'm going to show you some data from some old data from grape on that. The chemistry, and I think the company knows, but it's not something that I've ever been able to figure out. It's an ethanolic extract of the giant knotweed plant. Okay, so apparently there are extractives in the giant knotweed that can turn on this induced resistance. And as far as what it turns on, it appears to be salicylic acid, but there may be some turning on of JA as well. I'm just not sure, but it's kind of a mixture of things. And perhaps Barone Bioinnovations would be able to tell you if they were inclined. But it is certified for organic use as a reduced risk fungicide. I'm pretty sure it's certified by CCOF. I believe it is. Is that correct, Margaret? Is that?
I can double check, but
I think I know in other parts of the country it is. I know California has more restrictive rules on some things. So, which is basically produced from shrimp and crab shells. Which are very rich source of chitin. And chitosan is basically a breakdown product of chitin, which forms the shells. And you can with all that shrimp that we eat and that crab and stuff, what are you going to do all the shells? Well, this is one thing that they can do with it is produce chitosan. It has been around for a long time, over 40 years. At least we've known about chitosan as an elicitor in plants. In other words, eliciting this defense responses. Was developed originally by a colleague of mine at Washington State University many years ago. But then it's been commercialized, and there are various products. One that I have seen is called Beyond, and but it's had other iterations in the past, and so you can find this on the web. You can probably buy it on Amazon. And the active ingredient is chitosan, as I said, which is a carbohydrate polymer. And it's active against powdery mildews and soil-borne diseases. Its mode of action is, it probably mimics the action of other elicitors. See, certain fungi have a lot of chitin in their cell walls. And so we know from back in the past that during infection, some of that chitin can be recognized by plants such that you'll get a partial resistance to the infection as a result. And so that's what stimulated the interest in chitosan, this breakdown product. And it probably engages both the salicylic acid and the gismonic acid pathway to achieve its effects. And it meets USDA standards for organic agriculture use as an all natural plant amendment. So it meets it for a plant amendment. It can't, it's not really considered a pesticide in that regard. This shows you a comparison with the copper sulfate versus chitosan in seed, wheat seed that was inoculated. First, the seed was treated copper, and they were inoculated with Fusarium, the soil-borne pathogen Fusarium graminearum. And you can see that the difference in the stand, the non-treated control looks pretty lousy. The chitosan treated looks as good as or pretty good compared to the copper sulfate. That. Then there are aminobutyric acids, and this is GABA, BABA, and ABA, not to be confused with the aging Swedish rock group ABBA. So anyway, I know that's corny. So these are gamma, beta, and aminobutyric acids. So these are natural products, and by natural I mean they naturally occur in plants. So they're natural compounds. The most effective of the lot here is a beta aminobutyric acid, or BABA, and you can see what it can do in controlling sudden wilt of melon. This is from an old paper. Again, I'm a colleague of mine that I worked with a little bit. And you can see an untreated plot versus a plot that was treated with 2 soil applications at 14-day intervals. And so they look pretty well. So Baba seems to be, well, you don't see it here, but it's the most potent, it seems, of the 3. GABA was sold as Oxygro at one time. I don't know if it's still available. I haven't been able to really see it, but yeah, you could find it in the industry. There's another one I want to tell you a little bit about. It's something now called Axiom, and this is harpin proteins. What these are proteins, several proteins that have been purified from the cultures of the fire blight bacterium, Erwinia amylivera. Okay, this causes fire blight disease in apples and pears and other rosaceous species. And this also has been around for a long time. I mean, since the 80s. It's had, again, different iterations. Different companies kind of picked it up, and used to be called Messenger. It's now sold as Axiom by a different company, but it has activity in inducing resistance in grape to powdery mildew and botrytis bunch rot, and has shown efficacy in other diseases as well. It's kind of hard on some of these to really find good, solid efficacy data, but it is sold, and again, this is something that you can find on the web. So here's some data that were given to me by a late colleague and friend of mine, Doug Gubler, who some of you may have known, and this is old data on some work that they did with some of these different, comparing a whole slew of conventional fungicides as well as plant activators. And sulfur and so forth. So I just want to point, so this is on grape powdery mildew, and just looking at the berries. They also looked at foliar infection in these studies as well, but I just want to focus on the berries. So if you look at the untreated severity, disease severity, which and incidence. So the incidence is a more conservative assessment. Incidence simply is yes or no. Does it have powdery mildew lesion or not, and so that can be a little or it could be a lot, but severity really, I think, captures the impact of the disease much better than just incidence. So if you look at this column, they're all, I mean, some of these are conventional fungicides. There's sulfur, then messenger. Now we call it Axiom, the previous slide, and you can see that so they're putting it on 8 times, but they're also putting on these other materials 7 times during the season. Messenger again, but in combination with the strobilurin fungicide Abound, and you can see that it does even better, perhaps, does very well actually. Serenade, which is a biological bacillus from the species, and that but that in combination with Raleigh. Regalia, again, this is that extract from the giant knotweed, showed very good control. And then messenger, again, in combination with some of the best control is in combination with bond. And the reason I show you this is because now this would not be something you could do in an organic production. So you'd have to get rid of the Abound. I believe messenger actually, or Axiom, is registered for organic production, at least in some parts of the country. But what we've often seen with induced resistance, these plant activators, if they're used in combination with conventional fungicides, that seems to be where they have their best niche, if you will. And because using induced, it's a tricky proposition. Sometimes it works, sometimes it doesn't. And that's been really the issue with using induced resistance in production systems. Something that I want to mention is a product, a seaweed extract called Acadian. This is Acadian Plant Health. It's a company in Canada, Nova Scotia. And we've been working with Acadian, looking at their product. It's marketed, this is an extractive that they get from seaweed. And it is marketed as a biostimulant of plant development and growth. And so it's, there is a preservative-free version that is certified here in California for organic system. They have a preservative one, that version of it that is used in conventional systems, and actually it can be used elsewhere in the country for organic systems. Just that we're pretty strict here in California with respect to some of these products. So there is a preservative-free version, and what has been shown, so it's sold as a plant biostimulant, almost a fertilizer, but it does have some protective benefit against several fungal and fungal-like pathogens, fungal-like oomycetes. What really got us interested is they're very rich in these fish oil fatty acids, arachidonic acid and icosapentaenoic. You've seen the EPA on commercials for cardiovascular health and so on. Anyway, the seaweed is a rich source of these, and as well as certain carbohydrates called beta-glucan oligosaccharides. And both of these groups have been shown to be pretty potent elicitors. In fact, I started my career with discovering arachidonic acid and EPA as elicitors in these things. So we've been interested in this because they do protect against Phytophthora capsicum in tomato and pepper. And so we've been studying that mechanistically. And this happens to be some field trials from Acadian, I believe done in North Carolina, but in pepper. And you can see control here, and then Acadian in this plot here applied as a 1% soil drench at planting, and then 2 liters per acre by drip every 2 weeks. And so they just put it through a drip system there. So it protects against Phytophthora root and crown rot. These are fields that, an area that, a plot that has problem with that. So I'm going to finish up here and just say that there are costs and trade-offs in using it. And this is the tricky part about using induced resistance. So you can imagine plant defenses are costly. So if you trigger all these things, it comes at some expense. Now, priming, that is just setting the plant up so it's not completing things until the pathogen attacks it, can offset some of these costs. So in other words, doing it at the right time at the right place to complete the circuit can offset some of these costs, but it is expensive.
Rick, are you talking about energetically expensive?
Energetically, not expensive. Well, it depends on how much these things cost, dollar-wise too. And so you want to get the best bang for your buck, of course. But I mean, it's costly to the plant. Okay, is that clear? Yeah. And so it's because a lot of energy that the plant has to put into making these, doing this, what I say here, reorganization of gene expression. So what you see is this upregulation of defense genes and other things, but you see a downregulation of growth-associated photosynthetic genes. In other words, things the plant needs in order to fix carbon and grow. So you can, if you do too much of this, you can kind of put the plant at sort of get it stuck a little bit. There is some evidence for fitness costs. What I mean by that, the yield of induced plants, and this is reflective of what I just told you, under low disease pressure may have lower seed production than non-induced. So in other words, where you really see the benefit of these compounds is when you have some disease pressure. You want to be able to see the difference between but if there's no disease pressure, but you're triggering this induced resistance, what you can see in the induced plants is they're not going to yield as well. Okay, at least that's been shown experimentally. Now, all these products are a little different. The companies have looked at some of these aspects, and so it's important to read the label and pay attention to what is recommended. The other thing is something we studied about 25 years ago is that the salicylic acid-based resistance can increase vulnerability to certain insects, and vice versa. Too much JA in the plant can make the plants vulnerable to certain pathogens. So there's this trade-off that can go off. And that's why it can be kind of tricky to implement this in a production system. And then finally, just to say that the majority of the commercially available plant activators seem to target the salicylic acid induced defenses, although we're learning more and more about these things. And as time will tell, hopefully we'll see like chitosan where both pathways are induced. And I'll leave it there.